Thank you, Iris. Can you hear? Yeah. It's okay. All right, today I'm going to talk about studying the brain from the perspective of EE. And why study the brain? Well, it can do a lot of amazing things. And some of the things it can do are unfortunately beyond the grasp of EE. Like we would love to understand, for example, how the brain continues to work while it grows, how it recovers from injury, how it wires itself without any outside instructions. But none of those will translate very well into what we know how to do in electrical engineering. But there are some areas where the brain could really teach us some big lessons, because there's some things it does much, much better. And I'll talk about these. These are the subject of the talk. And just to give a couple examples, you look at power. Like you look at a little fruit fly that flies around. It flies around, it does optical navigation and flying, all with 125 nanowatts. If you look at the same chip in a drone that does the same task, it's a watt. Right? So it's 10 to the 7th times better. If you look at learning, a child learns a new language from a few million word examples. It takes the LLM's billions of words to do the same thing. So the learning works much, much better. Similarly, a teenager does incremental knowledge, I think maybe 20 hours to learn to drive. Think of how long it took to train all those Waymos, right? And all of that is about 20 watts of power. Your brain is about 20 watts. And so that's a fraction of one NVIDIA GPU. And from algorithms we can learn, because the brain does learn. It learns very well, but it doesn't use backpropagation. And probably that's the secret to how it does it so well. So if we can understand how the brain learns, there's a good chance we can actually use that in EE. Now the power consumption is particularly stark. Another example, a human drives, right? Their brain is doing all the work, 20 watts. This is an already trained system. This is inference and not learning, but 20 watts. A Waymo, same thing, 1,000 watts. And so right there's a factor of 50. And training is even more spectacular, because the human brain at the 20 watts is when it's doing inference. It's an extra 1 watt when it's doing specialized training. So if you're thinking really hard, you're using 21 watts. If you're just idling, you're 20 watts, right? So basically that teenage student learned the additional skill with a total of 20 watt hours. You compare that to what it takes to do even fine-tuning of an incremental model, it's crazy. Similarly, even if you take, for that same teenager, building the base model, right, all the way through teenage years, your brain has used a total consumption of about 3 megawatt hours. If you compare that to what it takes to train an LLM, I don't know the exact numbers for Waymo, of course, but training Llama took 21 gigawatt hours. So that's 7,000 times as much. Now, of course, the human can't speak 100 languages, so it's not quite equivalent, but the power consumption is pretty impressive. Now, this is a slide just for interest. This shows the power consumption of the human brain. It's measured by sugar consumption on the vertical axis and years of growth on the x-axis. And the top curve shows how much power your brain takes in terms of sugar intake. The bottom curve shows body mass. And so the brain peaks about age 5, but the body mass doesn't peak until you're an adult. And for some of us, it doesn't even peak then, right? We keep growing. But the amazing thing is, at the age of 5, your brain is using much more power than it uses now in a body that's a quarter the size. And in fact, when you're 5 years old, 60% of all the energy you eat goes into running your brain. So it's really, really important for humans. Now, a lot of EE concepts apply very well to the brain. So if you know EE, understanding the brain is easy. The top picture there on the right shows a gate. The bottom one shows a biological network. The two are pretty much the same. A computation is done by a network of elements. EEs call it a netlist. Biologists call it a connectome, but it's exactly the same thing. All the algorithms are implemented in the same way, just as activity propagating through the network. It has very similar attributes like stuck-at faults and power consumption and so on. So if you understand EE, learning biology is not so hard. The fundamental computation unit in biology is a neuron in your brain. And if you look at the neurons, like in the pictures here, they look like they're wildly different, but they're not. They're all doing the same operation. The only difference in that shape is who their inputs and their outputs are. So really the shape things are all for figuring out connectivity. And they only do one nonlinear operation. And that nonlinear operation is summarized as integrate and fire. And if you look at this picture here on the left, that's the equivalent circuit. So what's happening is The neuron is insulated by its cell wall. So inside it has a voltage. When synapses come in from other cells, they can cause current in, which raises the voltage, or current out, which lowers the voltage. And of course, there's always some leakage across the membrane, which sort of stabilizes it out. If the voltage rises to a certain amount, it triggers a positive feedback loop that then drives the thing positive towards the top rail. That triggers a negative reset mechanism that brings it back down. So what happens is you get a spike. That's what a neural spike is. It's just an integrated fire model. And if you look at the pictures down below, the second one in the middle shows what happens with increasing current. As current goes up, as shown in the second line in the middle, then the frequency of firing goes up, as shown on the bottom. And if you draw that in the frequency domain, the plot on the right has frequency on the Y and input current on the left. You see you get nothing at all until you reach the threshold, and then the firing rate is proportional to the input. And this is exactly the ReLU unit that's used in training machine learning, which may not be a coincidence. So it's doing basically the same operation on every neuron. There's also gap junctions, which I won't cover much here. These are simple resistors between cells. They make a little hole in the cell wall. Current can go back and forth. It gives you always a positive sign and gain of less than 1, but it's used a lot in insects. It's used in your optical system. It's not used much in the center of the brain. Now, the neurons are also the wires, right? We don't have gates and wires. We have one thing that does both. But neurons are really crappy wires by biology standards. If you work out the numbers, right, if you have I have the numbers here for resistance and capacitance, but salt water is not a very good conductor. It's about 10 to the 8th times worse than water. And if you work out the numbers for a 10 centimeter long connection that goes across your brain, the propagation time is about 1,000 seconds, which is 16 minutes. And so clearly this is not how biology works, right? You can think faster than that. And the way they do it is pretty familiar to double E's. What they do, if you look at the top, the nerve actually has channels all along it. And each part, as the positive voltage appears here, flips the adjacent channel to cause positive feedback there, and that sort of increments along the nerve, just like a chain of dominoes. You see a chain of dominoes falling like that? It's exactly the same thing. And the equivalent electrical circuit is just inverters stacked end to end, like you see over there. And that gives you a much higher speed. It's sort of the way we would do it. And that will reduce the time across your brain to on the order of a second. So that's pretty good. That's better. But we can do even better than that, as we know, which is that for a sufficiently short period, a short length, the propagation of the electrical potential is faster than a single gate operation. And so rather than chaining gate operations together to get a long distance, what you can do is you can propagate a little ways by electrical, then use a gate, a little ways by electrical, and then use a gate. This is exactly like repeater insertion in VLSI. It's exactly the same. And biology has come up with exactly the same solution. So this picture on the bottom, they have these little insulated segments with little repeaters in between them. Looks like this. So if you look at it close up, the individual sections are insulated by this thick yellow thing over here. It's what we call myelin. And then this portion triggers, then it propagates electrically to the next set. Then the next set triggers electrically to the next set. And with that, you can get speeds up to 150 meters per second. And so it's 5 or 6 orders of magnitude faster than the basic operation. And like I said, this is obvious to double E's. But the thing you can notice about this is the insulating part is white, and the computing part is gray. And that's why when you look at the brain, you hear about white matter and gray matter. Well, white matter is the wires, and gray matter is the computation. And so you can even see that in the brain. You can see the channels between the parts and so on. Now, just like double E, that means when the brain does a computation, there's 2 regimes. If the computation can be performed in a small area, then you don't need repeaters. The cells are basically isopotential, and the computation occurs just like a series of gates. In anything big, like between the sides of your brain, just like double E, communication delays dominate. They're far bigger than the gate delays once the distance gets bigger than about 100 microns. So the big distances, it's wires. Small distances, it's gates. Now, if you want to look at this in detail, these kind of calculations, biology has exactly the equivalent of SPICE. Spice is what you use when you want to look at all the details and you're not worried about speed. The exact equivalent program in biology is called Neuron, and it uses exactly the same techniques. It takes a big cell like you might have, divides it up into small discrete compartments, generates the differential equations with diffusion and current sources and everything else, and solves them with matrix algebra. And the matrix problems it solves are exactly the same as Spice, which is a wide variety of time constants, uses the same matrix techniques and so on. So this is another area where EEs and biologists are actually pretty similar. And just like EE, when you have these really complicated systems, you can use model order reduction. And so there's no real reason to take one of these giant neurons and divide it into 10,000 compartments. You can come up with a much simpler model that does exactly the same thing. And just as an example, here's 2 papers written in different fields, written by different people, containing exactly the same ideas. That's easy to do. And the next one that's similar between biology and EE is statistical timing. It turns out in biology, everything is statistical. They're running very close to the thermal threshold. Everything varies. And if you look at the currents, or you look at the measurements, you'll see perhaps over here on the right, this is measuring the current into a cell. And you can see the current varying as the channel opens and closes due to thermal variations. You can similarly draw diagrams like on the right. That's a diagram that's talking about the conformation of molecules in a complex. And they switch configurations from one to another, and they switch at rates determined by these various rate constants, and that determines whether the channels are open or closed. So this is exactly like Markov models. Not your Markov models, but the other Markov models. And in biology, almost every action is statistical because the numbers are small. For example, when a neuron releases a neurotransmitter, it only releases about 5,000 molecules. And what that means is your standard deviation will be somewhere around 70. And so your standard deviation is 1/70th or so of your value. Likewise, each one of those diffuses and then triggers a few ion channels, but it does that statistically because diffusion isn't uniform and so on. And those trigger, and then they relax due to thermal constraints, so they relax on a statistical timescale also. And so EE has looked at this for years. There's a whole literature, Sachin, a number of people here have worked on this, about statistical timing. This has never been applied to biology. I think there's a PhD thesis just waiting to be written where you could take these techniques from EE and apply them to biology. Now, the next one we'll look at is power. Right? As we saw that biology is often 100 or 1,000 or 10,000 times less power to do the same job. How does it do that? Well, there's 2 components, hardware and software, basically. So the first component is hardware. We know, for example, that voltage goes like V squared. Right? That's why we run as low voltage as we can. Now, if you look at the equation for a MOSFET, you see this E to the KT over Q in the current term. And what that means, if you want to turn it all the way on or all the way off, a 1,000,000 to 1 ratio between on and off, you need 14 KT over Qs, which is about 1/2 volt. And that's why we can't run the voltage for a MOSFET much below 1/2 volt, because it won't give us enough on to off. On the other hand, biology does exactly the same thing, but they do it with 50 millivolts, 10 times less. So how do they do that? Here's a picture of that, by the way. On the right, we have a MOSFET. X-axis is voltage, y-axis is current. You can see it takes about 1/2 volt to turn it all the way from on to off. If you look at the one on the right over there, that's the same for an ion channel in biology. And you can see that 50 millivolts is plenty to turn it all the way between on and off. And so that's why they can use low voltages. So how do they do that? Well, in biology, all the currents, or most of the currents, are dominated by these things called ion channels. And these are holes in the membrane that let positive or negative ions in and out. And they're triggered by all sorts of things. They're triggered by voltage, they're triggered by mechanical stress, they're triggered by molecules that bind to them, and so on. So they're all built like this. And one thing they have in common that you'll see is they're all 5 or 6-fold assemblies of molecules. And that's super important because what's happening here is all 5 of these are gathered around a hole, a pore, and the pore only opens when all 5 are in the correct position. Now, each of them itself is going to be subject to this KT over Q thermal problem, right? It can't always stay in the right position. But if all 5 need to be open, now you need, statistically, 5 times the e to the 5 kt over q, not kt over q. And now, for every 27 millivolts, 1 kt over q, instead of getting an e-fold change, you get a 148-fold change. And so 2 times that, 50 millivolts, you can get your 100,000 or a million on-off ratio with just 50 millivolts. And so it's all done by these fact that they have these 5-fold symmetries in these ion pores. By the way, if there's any questions or anything, just raise your hand. We can stop and do them while we're at it. Of course, now that's only a factor of 100, right? That's not the whole factor. So there's also from processing efficiency. For example, the clock speed is much, much less. When you are driving your car around, you're processing at roughly a kilohertz rate, not the gigahertz rate of the processor in a computer. And so you're doing the same task, but with 1,000,000-fold less clock cycles. And so if we can understand how we did that, now that's something we could really use in terms of EE. We can turn this kind of understanding into improved systems. Now, how does it do that? Well, we don't quite know, but we do know that the circuits are very different. For example, if you look at what's the fan in and fan out, typically in a human system it's 4. In a biology system it's typically 150, 200, even 1,000. So the fan outs are much, much larger, and the paths are much, much shorter. And the shorter paths are illustrated in this plot here. What we show on the y-axis is how many hops it takes to get from one gate to any other gate, where that's 100% of the other gates, and that's none. On the x-axis is how many hops are needed to get that percentage of gates. And what you can see is these are all biology systems over here on the right. You can get almost anywhere you want in 3 hops. If you look at man-made systems, then you see that curve on the left. That's one of the old benchmarks. But the answer is about 80 to 100 hops to get to just sort of saturate the system. So the biology uses much, much shorter paths with much, much wider fan-out. And that's probably a key to how it can do it in so many fewer clock cycles than the human stuff. So here's a biological neural net. You'll notice that there's a couple differences between this, a machine learning neural net, and EE. One is it's got relatively few layers, typically 3. This is in the eye. It's not neatly organized, right? There's no particular pattern here. It's got lots of lateral and feedback connections, and that's interesting because people don't tend to put these in EE circuits without latches. But there are no latches here, right? They're building these recurrent circuits. It's got no clocks, and it's designed by evolution, and we don't know how it's trained, right? We don't understand how the training algorithms work. But it's fairly different than the human ones. So now understanding these, I think, is likely to result in huge savings in terms of what we can do with electrical engineering, right? How would you design a network to do something like this? We don't know, right? How do they work? That's also hard. It's very similar to the problem of looking inside a machine learning box and figuring out how they make their decisions, right? We know that they work. They were designed to work. We don't know how they work. Now, interestingly enough, for the brain, we do know how a lot of the circuits work, but they're all reflexes, because reflexes have to be fast. They're not trained. So those ones we know. How general learning works, we don't. How are the weights determined? How are the network trained? And, very interesting, how could you synthesize these circuits? Right, this is something our synthesis tools don't even try, is to build circuits that have loops and feedback, but no clocks. Now, when you look inside the brain, one thing that looks complicated, but isn't, is this large number of cell types. And like I said, the difference between the cells is really just how they're wired. They're not doing any different operation. And so the cell type thing is sort of irrelevant to us as EEs, except it provides a very convenient means of analyzing the circuit, which is we have all these cell types. Each of them has a certain set of genes that are turned on. You can insert special markers in those genes that force the neuron on, force the neuron off, force it to respond to light, and things like that. And so you can create your own stuck-at faults. And that's how we go about understanding the brain. And so you can take exactly what we do from test and put it into biology, and that's how people figure out how these neural circuits work. Okay, so far, everything I talked about is a direct analogy between EE and biology. It's pretty easy, right? You have neurons are gates, ion channels are synapses, capacitance is the same, thresholds are the same, signal propagation is like wire delay and repeaters, gap junctions are resistive coupling, it's all similar stuff. But there's an extra signaling element in the brain, which is something called neuromodulators. And these work more like global variables, that instead of just talking to the adjacent signal, or adjacent net like this, they soak through the thing and they talk to people who are far away. So they give you the idea of global connections. And it's well known. Here's, for example, of 3 chemicals that are known to affect the brain. We already had experience with these. Like last night, you probably drank some ethanol, which you see there on the right. In the morning, that gave you a headache, so you took some aspirin over here. And then you got here and you were tired, so you took some caffeine. Right? So these are all well-known stuff. Now, these are ones that affect the whole brain. Lots of others only affect parts of the brain. They're very common. It looks like, from what we know, about 20% of neurons express these neuromodulators, and probably a higher percentage respond to them. They behave very differently than the direct connections between neurons. If you look at this chart here, on the y-axis is the peak concentration from a release of 1 vesicle's worth of neurotransmitter, neuromodulator. On the x-axis is the distance. And you can see the peak concentration goes down as the distance gets higher. The 2 dotted lines are 2 different sensitivities of receptors, a weak receptor and a strong receptor. And what you can see is that first vertical line, everything for a weak receptor, it affects everything out to a 2 micron distance. For a more sensitive receptor, it affects everything out to a 7 or 8 micron distance. And those are pretty big. If you look at them on the chart, here's 2 microns, that's maybe the nearest 100 cells are affected. And here's 7 microns. You can see this is going to affect the nearest several thousand cells. So these are variables of a very different type. Also, they're relatively slow compared to the direct connection synapses. If you look at this chart here, on the x-axis is the distance. On the y-axis are 2 curves showing the on time and the off time. So you can see if you're close, like 1 micron, the on time is almost instant, less than a millisecond, but the off time is 100 milliseconds. That's a very long-lasting pulse. If you're 8 microns away, like on the right, what you see is it may take you 20 milliseconds to turn on, and at 40 milliseconds, you'll turn off. So although these things can talk to a lot of people, they're quite slow. So can we model these? Yeah, sure. What we need to know is who gives them off, who can receive them. We need to add them to our connectome. We need to know how much is released. We need to know how it diffuses in the tissue. And we need to know how sensitive the receptors are. Now, in theory, all this information can be gained by going through the genome and looking for receptors and transmitters and so on. And there are people working on this actively now. But it's a lot of information we don't have. You need different data structures. I won't talk about this. And the diffusion equations are pretty well known. Now, this is a traditional diffusion equation with a proportional term in there. This equation has been extremely well studied because it's used in all sorts of sciences. For example, if K is positive, that's the equation that dominates the atomic bomb. If K is negative, you get diffusion in the brain. It's the same equation, used for 2 very, very, very different purposes. But it's well understood. Now, when you solve for these, what you see is that you get 1 release, it propagates out, at distances out, you get a big pulse, and you turn on and off. But you get some problems that I'll show here. These are problems that also occur in logic simulation. But say we have in this chart, we show the concentration on the Y and the time on the X, and the concentration versus various times. Now, if we start with this, let me see. Get this on. Okay, well, that's okay. We start with the first event. What we'll see is now we have to schedule 10 milliseconds later an on event and an off event at all these other neurons that are out there. But if during that 10 milliseconds we get another event, indicated by the second, okay, there's our first release. That generates those 2 potential events out in the future. So those are scheduled, just like in a logic simulator. Now, if while we're waiting for that event, we get another event, now what happens is the concentrations of the transmitters add. And now the terms become different. And so we have to take those events that have not occurred yet, once we add those up, and change them. And so what we do is it's just like a logic simulator. We have a queue of upcoming events, but they're only tentative until we get to that time. Until we get to that time, other releases can happen and move the on time in and the off time out. And so fortunately, all this stuff is understood because it happens in EE. Here's a plot of a standard TTL mux thing. And what you see is the paths that go to the output are of different lengths. And so exactly the same problem happens. An input comes in at one of the slow inputs, you schedule the output, but before that happens, one of the fast inputs changes, and now you have to change your mind. And so logic simulator people have dealt with this for years. And so we can use exactly the same technology to deal with the EE. So on the other hand, some calculations are still analog. You can get a lot of small responses that add up to which do nothing by themselves, but add up to a response that actually happens. And so that's something that doesn't happen in digital logic. So for neuromodulators, basically, we can add these. None of our current simulations cover neuromodulators. We think we can do it. We have prototypes that can do it. We need a lot of experimental data. We need new models. And we need both static and dynamic analysis. This is very similar to EEs. Okay. So now where to find out more about this topic? There's a lot of books and other things. I'll talk about those, and a lot of websites. This is perhaps the classic textbook in the field. It tries to talk about all of neural science, and as a result, you can see the page count varies almost linearly with time. Right? The book started out this size, and now it's like this big thick, and pretty soon it'll be a 2-volume set. If you want to look at it from the point of view of EE, there's this book from MIT, which is great. It's written in very EE language. It has all the equations. It discusses the limitations of the model, stochastic behavior, and so on. So this is the one, if you're a EE, this is the one I'd recommend to read. So where are we now after all this time? Well, we have the complete synaptic circuit for some small animals. For example, we start with the worm. That's 302 neurons. We also have the larva of Drosophila, maybe 3,000 neurons. We have an adult fruit fly, that's the fly in the picture there, that has about 160,000 neurons and about 300 million synapses. And we recently released results that have both the brain and the spinal cord connected, because it turns out a lot of the calculations of things like walking and moving and flying happens in the spinal cord. It's not done in the brain, so you really need both. People have built these combined neural physical models, and they can sort of have flies staggering around. They're not accurate yet, but they're being done. And like I said, we can do stuck-at faults and things to verify our models to see how well they work. So far, all of these have been obtained by electron microscopy through a very painstaking process. We take a fruit fly, it's less than 1 millimeter thick. We cut it into 100,000 sections. And so then each of those we image with a big electron microscope. We stack them together, it's several terabytes of data. And then we try to reconstruct everything in 3D. It takes a lot of machine learning, and then that's not accurate enough, so you have to correct it by hand. So it's a real tedious process. Even these small animals here that you see took many years and many millions of dollars to reconstruct. And if you look at the mouse brain, we'd like to do the mouse next. Mouse is 10,000 times bigger, and the human is 1 million times bigger, right? So we're clearly not to the point of being able to do those yet. So there's a new technology, which is one of these things I would have sworn would never work. What they do is they take the brain and they embed it in a gel, which is derived from disposable diapers, believe it or not. And then they take that and they soak it in water, and the gel expands uniformly. And it generates a very transparent and expanded version of the brain. And you can do this by big factors. Like they have people working at like factors of 20 or so. And that's enough to turn a 10 nanometer thing, which you could only see in EM, into a 200 nanometer thing, which you can see in an optical microscope. And so once you can do that, the potential is huge for faster improvements. For example, it's much cheaper, right? A fancy optical microscope is $250K. A fancy EM is $5 million, right? So big savings in cost. It's much faster because to work your way through the sample, you just need to change the focus plane. These expanded things are very nearly transparent, and you can see all the way through them. And so you just need to change the plane of focus, not physically cut it like you do with a EM. And finally, they can do multicolors. You can label all the components using antibodies and dyes and things. And so you can see exactly what's going on. And that's a big help, because right now we have to guess at things because we get these monochrome images, and it's hard to tell neurotransmitter identities and things like that. Hopefully with this technique we can see them. I'm going to skip the details in the interest of time. So where are we? So overall, there are some things we understand pretty well. These are reflexes. These are hardwired, escape response, and things like that. That we understand. There's some sorts of conditioned learning we understand. Like the fly can learn to like this odor and not like that odor. We know how that works. That's a built-in network in the fly. It's just changing the weights. We know how the weights are adjusted even more or less, so we understand that. Now the neuromodulators, the chemicals that soak through the brain, we don't know those much at all. We don't know how many there are. We don't know where they act. We don't know how they interact. And unfortunately, that's the thing that determines sort of motivation level stuff. It's slower than reflexes. That would be things like fright and food and so on, but we don't understand those. And we definitely do not understand the sophisticated kind of learning that happens in humans and other big animals. No backpropagation, but we don't know how it works. So where does the future hold for this, right? It's very promising. Electrical engineers can help this a lot because the basic operation of the brain is electrical. And so all of us can help biologists. That's sort of what I moved into the field to do 20 years ago. It turned out at that time we didn't actually have any circuits. So I sort of detoured into trying to get the circuits. We have some small brains, worms and flies, and we're working our way up. We're going to need better techniques like statistical timing, things like that. So people who understand stuff here can help. And we need theorists. So, and by understanding these things, the benefits are potentially huge. You know, mostly power, speed, and learning methods and results. And so I would argue that if you're a double E and you're looking for a change, come and study the brain for a change. And so with that, I'll open up for questions, and thanks very much.
Thanks very much for the inspiring talk. So we are open for questions. So,
thank you, Lou, for the great talk. At some point, you said that there's no clock in these networks, right? They're not clocked.
No, they're not clocked.
But most types of communication, including human speech, need clocks.
Yeah.
Motion, physical motion, needs clocks. So there's something missing here.
There's definitely a time axis and time varying behavior, but there's not the regular clock like we wait for this before we do that kind of thing. And so how it gets determined, I don't know. Maybe it's some multiple of the fundamental time constants. Maybe if you built a mechanical copy of the brain, you'd talk 10 times as fast. I don't know, but there's a definite time component, but it's not the way we do it in electrical circuits. There's no explicit synchronization between the steps. I'm not sure if that answers your question.
Well, it's pretty amazing that things work.
Yeah, it's amazing it works at all, but of course it doesn't, right? It's thought that things like epilepsy and things like that are failures of the timing system, or where the system, because it's built like this, can become overstimulated and drive itself into positive feedback loops. And so it's like anything else in biology, it's probably running right at the edge of stability. Because if you're not running right at the edge, you're not running fast enough. And so probably that's why your brain is a constant temperature. Because if you have to take a temperature variation, that means you have to back off from the optimal operation to ensure it works even in the high temperature case. So by keeping constant temperature, that's a very expensive thing to keep constant temperature. And so for sure that has some important reason, and probably to keep the timing under control, I would guess.
Yeah, makes sense.
So a question about neuromodulation. Could you think about it as dynamic voltage scaling or noise in the supply network that affects gates within a certain region?
Dynamic voltage, that's a good question. We don't know. It triggers all sorts of receptors in the other cells. And unfortunately, most of them are what are called G-coupled receptors, which means the ligand binds on the outside, and what that does is it goes inside and starts to change the way that DNA is transcribed into proteins. And so it can have almost any effect at all. There's almost no way to predict what they do. G-coupled receptors drive all sorts of stuff. And so it's very hard to predict. It's very hard to say that neuromodulation will cause one specific effect. Also, there's several hundred chemicals, and they're all diffusing at the same time. And each of them has a different role, we think. And so it's pretty hard to make a general statement about equivalence.
If I can ask one more question. You talked about expansion microscopy, and it seems to distort the shape of the object that you're looking at, and I assume the distortion is not isotropic. So can you reconstruct the original from what you see?
Yeah, that's a good question. The question is, it's not isotropic, and it's true. It's not completely isotropic, but of course the connection list is all topological, right? You can bend the thing arbitrarily, and the same things are still next to each other that were next to each other before. And so for that purpose, for the purpose of synapses, it makes no difference at all. For the purpose of neuromodulation, now we're distance dependent, and it would have some effect, but it's surprisingly isotropic. It's actually, like I said, I'm amazed that it works at all.
Even in the third dimension?
Even what?
Even in the third dimension, height?
Yeah. The things are quite light, and the overall thing looks like a little piece of cloud. But a little 1 millimeter thing gets expanded out to a 25 millimeter on a side cube, and then you can image the whole thing. It's astonishing. I would never have thought it would work.
So I have a question about the variability. If you take the chips in your cell phone, the schematics are the same, right?
Yeah.
There's some process variability, but
Yeah.
that's manufacturing, right?
Yeah.
So if you take two different individual of the fly, will the connectivity be exactly the same, or does it vary?
No, it's definitely not exactly the same, because there's a couple of things that affect that in the fly. One is how much nutrition it has, because it sort of grows to some fixed point, and it's forming connections as it goes, and it stops when it runs out of food, right? So if they grow up in low nutrition conditions, they're definitely smaller circuits. The second is some of it is deliberately variable. We know that in the olfactory system, there's 60 receptors that come in that can smell 60 basic odors. And what it does is each side of the brain, in fact, is different. It builds a hash function. The synapses in there connect at random to a subset of these things and generate several thousand hashed values from the combinations of odors. And then these project through a PLA-like structure that, that when you get a stimulus comes in, it looks it up in the PLA and decides on the response. And those are adjusted by weights. But those are completely random, or almost completely random, like any good hash function. And they're in fact different, and the left half and the right half of a fly brain are different. So some of them are deliberately random, some are accidentally random. One thing is we don't know about that as much as we should, because these things are so expensive to reconstruct, we've only done one of each. So we don't really understand the variability.
Is there a good understanding of what they have in common? Obviously a lot, but not everything. Where's the boundary?
Well, we don't know. We do know we have done one male and one female, and those differ by about 10%. So 90% of the circuitry is precisely common between the male and female, and the other 10%, some of it we understand, like there's no egg-laying circuit in the male. Some of it we don't understand. It looks like maybe the aggression circuits are different, right? And the trade-off between aggression and feeding and things is different in the male and the female and so on. But because the numbers we've reconstructed are so small, this is very much a topic that's not well understood, is inner animal variation.
Hi, Lou. Thanks for another great talk. I have two questions. Yeah. One is both thinking back a ways, but there was this thing called a brain map project, and obviously at the scales you describe, that seems very far out of reach today. Although it was launched many years ago. Can you say something about sort of that?
Actually, when we refer to connectome, there's actually two meanings of connectome. There are people who do like this, which is a really detailed study, neuron by neuron, of a small brain. There are also people who look at the overall connectivity of things like a human brain using techniques like fMRI and CAT scans and things like that. And they produce a brain map, but that's just a map of region to region. Okay. So their regions are on the order of a millimeter cubed. And that, for us, would be a huge area, right? But still a lot of those in a human brain. And so there's two different scales, and they're way different. And the one that was called brain mapping was the big scale one.
Okay.
And not the small scale one.
And the second question, when I was a grad student, people were talking a lot about artificial brains, constructing artificial brains, and they talked about the hippocampus and holographic memory and things like that. So is that still a quest?
I think...
For example, I mean, plasticity, holographic memory, realizing that in materials that we work with and manufacture, as opposed to, you know, cloning people.
To my understanding, there's not so much effort in that. It's sort of split into two areas. One is people building these huge neural nets out of stuff we understand, these giant layers and stuff, and then people trying to understand how the biology works. There's nobody sort of in the middle trying to build a biology-ish system that can do the things. I think they've wisely given up until we understand how the biology works, or until we know how it works a little better. But I haven't seen much research in that area. But that's just me.
So, Patrick?
Can you talk a little bit about the Elon Musk effect of the brain-machine interface or brain electronics interfaces? Can they be made reliably now, or what's the latest state of the art there?
That's the interesting question of these people who are working on brain interfaces. And what they do is they put a few thousand electrodes in the brain, one way or another. I think Musk uses tiny wires, and the one that we build at Genoa has a little chip that's embedded with little electrodes on it. And the question is, can you decode enough from the signals you can measure, the 1,000 or so, to do something useful? And there are people working very, very hard on that, because of course it would be great for paraplegics and things like that. And of course it might be good for just, you know, direct connection to a computer, so you wouldn't need to type or something like that. It's made some progress in terms of the replacement of existing functions like sight and things like that. The driving is not so good. The receiving is pretty good. And so whether 1,000 electrodes is enough, I think is a technical problem. I think for sure it'll work once you get to 10 or 20 or 30,000 electrodes.
Does it continue to work? I mean, so that sometimes the connection will be rejected by
Yeah, so the question was, does it continue to work? And that's a problem. We have had some that work for years. It's not quite understood why. Many times the body rejects it and starts to form a scar tissue over the electrodes or the wires, and then the thing stops to work. And that's been extremely depressing for some of these people who had their sight restored, and then a month or two later it goes away. And that's caused a lot of trouble with the ethics boards for approving these experiments with humans if the effect is known to be temporary and there's no permanent improvement. People are, of course, working on methods that would keep better, keep less rejection from happening. I don't think that's very well understood yet. And if I was Musk, I would be spending more effort on that and less effort on some other things, because I think that's a very fundamental problem in the field.
Thank you so much. So I have a short question. Sure. If one day we can totally simulate the brains, so how it would affect the world of fields, specifically for AI? Yeah.
That's a good question. Yeah, I mean, it's not completely hypothetical. We can do that now with the fly. We have the full nervous system and the spinal cord, and we can simulate flies flying and flapping their wings and everything with mechanical simulations coupled to neural simulations. So it's possible for flies. Now, the mouse or the human is a much bigger scale, and even if we can get Moore's law-like progress, we're probably talking 30, 40 more years until anything practical could be done. And also, all the methods now require destroying the brain, which people might be reluctant to do unless they're sure it's going to work. And so, once that happens, what are the effects? That's very up in the air, right? Nobody really knows. Of course, the rich people will get this first, and so it's probably not good. But other than that, no one knows what the effects are.
Okay, thank you so much. So I think our SPD organization prepares some gift to our distinguished keynote speaker.
Yeah, so we have also a little gift for you, and I'm looking forward to the day when asynchronous circuits might beat us in the game of memory. I mean, for digital, I'm pretty sure they would beat us already. But among the many things that I messed up was to hand gifts to all the keynote speakers. I see that Leon is still there. Let me briefly, and I heard that Leon already has his memory, so you have to solve a puzzle then instead.
Okay. Appreciate it. Okay, hey, thank you very much.
You're welcome. And I don't see Prit. Are you still here? Is there any colleague of Prit who works close to his lab? Or Igor? Can you take something for Prit? Okay, I will hand it to you, but then maybe we close this session with a big thank you.
Yes. So let's thank our distinguished speaker again. Okay, thank you so much. And